In this lecture, we're going to look at a different problem from aligning homologous sequences, which is to search for homologous sequences in a database. So we're going to see this problem of searching database and how this differs from aligning sequences that we assume to be homologous. We're going to look at two different search algorithms, FASTA and BLAST, and some aspects such as the statistical significance of the results we find, efficiency in the search, and so on. And there are many heuristics here. So one big difference from the previous algorithms that we saw is that in this case we are not trying to guarantee an optimal solution like what we saw with the sequence alignment algorithms, but we are using heuristics, so tricks, so to speak, in order to speed up the obtaining the results, speed up the computation and the search for results. So without guaranteeing the optimum, we can have something in a feasible time. We're going to look also at iterative BLAST, so PSI-BLAST, to enable us to find more distant relations between the query sequence and what we find on the database, and also look at other sequence comparison methods that do not depend on computing sequence alignment. This lecture will be a bit different from previous ones in that I'm not going to detail these algorithms too much, so we will still be focusing on the practical parts, on the implementation of the previous algorithms we saw, the sequence alignment algorithms. For these, I'm only going to talk about them a bit more superficially while we focus on the others in more details. So let's start with this problem of sequence search. One aspect of this is that in order to search for homologous sequences in a database, we will need to do many sequence comparisons. For example, GenBank has 230 million sequences. With the whole genome sequencing programs, those amount to 1.5 billion sequences. So even if we can align two sequences in fractions of a second, we can need months of computation just to search through all of these sequences in these databases in order to find a match for a single sequence that we have as a query. So even though those alignment algorithms using dynamic programming that we saw are fast in the sense that they allow us to obtain the desired results, the optimal alignment in a small amount of time, we need much faster algorithms in order to be able to do the same thing millions and millions of times to search a database. Another difference is that we are not assuming the sequences to be homologous like we do when we align them. If we have sequences that have some homology, we will use alignment to try to find the matches of the residues. When we are searching on a database, we are going to go through many different sequences that have little or no homology to the one we are using as a query. So in this case, we will need to have some measure, some statistical measure of how likely it is that by chance we can have some particular score so that we can discard those matches that occur by chance and find those that are statistically significant. Also, one thing, in the case of the alignment, we can either use global alignment or local alignment depending on what we want to find. If we want to find the best matching subsequences, we would use local alignment. If you want to match the whole sequences, then we use global alignment. But in the case of sequence search, global alignment is not a good option because often we can find good homologies in other sequences, but only in parts of the sequences. This is mostly because proteins, and so genes too, are modular in nature. Proteins fold in well-organized domains, but one protein chain can have several domains, and it can share specific domains with other proteins. So local alignment is best for sequence search. Now, there is a big difference between searching and aligning in that with alignment we assume the sequences we are aligning are homologous. So we are going to try to find which regions in the sequence, in these sequences, come from the same regions in the ancestor, and do that by assuming that homology can be inferred from the alignment. So we use the alignment between the sequences that we have to try to infer something about how they evolved into those differences. When we are searching, we cannot assume homology because we are trying to find which sequences in the database are homologous to the one we are using as query. So most of them will not be, and we need to distinguish between those. And so we here are trying to estimate which sequences are homologous, and we also need to do this very quickly because we need to search through very large numbers of sequences in order to find the ones that we want. So in general, if you use these sequence search algorithms, you may have in the end something that is an alignment. You have the representation of the match between your query sequence and the ones in the database that were found to be homologous, but this is not a good alignment. So it's not an optimal alignment. If you want to really obtain a good alignment after you get the results, you need to use those algorithms that we saw before, like the Smith-Waterman or the Needleman-Wunsch, depending on the alignment you want, to obtain a good alignment between those sequences. So remember that the algorithms we're going to use for sequence search are not really good alignment algorithms. They use quick and dirty methods, so to speak, in order to find the solutions in a feasible amount of time. So let's start by looking at FASTA. This was the first one widely used for sequence searches. It's still widely used mostly for DNA, and the way it works is that it starts by finding identical words of a certain size. In the case of proteins, these words are consecutive sequences of symbols. So in the case of proteins, we have words of 2 amino acid residues. In the case of DNA, it's 4 to 6 nucleotides. So the reason why fewer are needed for proteins is that in proteins we have 20 different amino acid residues. So combinations of 2 already only appear once in 400 in general. So they are sufficiently rare to be meaningful. Whereas for DNA, we only have 4 different symbols. So if we were going to look at combinations of just 2, this would be too common just by chance to be informative. So what FASTA does is that it finds these matches in these small subsequences of identical sequences of symbols, so 2 for proteins and 4 to 6 for DNA, and then it's going to expand this by looking at similar at similarity scores, so at similar substitutions. Basically, these similar symbols are those that have still a positive value on the substitution matrix. So it's likely to find them substituted one for the other. In the case of proteins, there are many amino acid residues that are chemically similar and often are easy to replace in the protein without having a large impact, for example. So starting with these cores of identical subsequences, FASTA will now expand by looking at similar residues in the neighborhood, and then join these segments and use dynamic programming for alignment, but only within a small number of possible displacements between the 2 sequences. So basically, graphically, it would look like this. If we represent one sequence in one direction, for example, the columns is one of the sequence, the rows are another sequence, and we represent a dot wherever we find identity between the residues, we can see here small words, small stretches of each sequence where the two sequences are identical. So the FASTA can start from here and then expand this, join those sequences that are close together, and then do the alignment only on this region of the matrix. So instead of having to compute all the possible combinations, it does the alignment only around a small threshold. For example, 32 positions in width. This means that the sequence alignment step is much faster because we are not computing the whole matrix, only that diagonal part, that lane here. And this way we can have one alignment between the two sequences very quickly. However, because we are restricting the sequence alignment step to only this region, we cannot guarantee that we have the best alignment. This can be something that is considerably below what the best alignment score could be. So this is very fast because it can index these words previously on the database, so it knows which sequences have which words, and then this dynamic programming step is also fast because it's restricted to only this small region of the total number of possibilities for aligning the two different sequences. So another thing that became standard with the FASTA algorithm is the file format. This FASTA file format is very used for storing sequence information and has a first line starting with this greater than sign. This line has a description of the sequence, and then it has a number of lines with the characters for the sequence, whether DNA or protein, but you can have split into several lines the sequence. And these files can have several entries for different sequences. So every time we find this special character, this indicates that it's a description line, and then this line is followed by the actual sequence. So this is a file format that became standard, and it began with the FASTA program, so it's still called the FASTA file format. Another algorithm is BLAST. This is more recent than FASTA, and it is a lot faster than FASTA. So in general, for proteins, this one is better, especially if we are looking for close local matches. For DNA, FASTA is still commonly used. And the reason why BLAST is much faster is that it completely skips the alignment part. So that part of aligning using dynamic programming that FASTA does quickly because it restricts to only part of the combinations, part of the matrix, BLAST completely ignores and focuses only on increasing or creating these maximal segment pairs, which is the sub regions of the sequences that are the best match. So it's looking for this local alignment and finding the pairs of segments in the regions that are more closely matched together. And basically BLAST also starts with this index of words, but unlike FASTA, it's not going to find the matches by identity but by similarity. So it has larger words indexed, and it will match our query sequence with sequences in the database according to those indexed words. And then it's going to expand and join these segments, these matching words, in order to find these maximal segment pairs, which are subsequences that maximize the local alignment there. So in a diagram, we have this database with lots of sequences. In all of these sequences, we have previously indexed which sequences of K symbols occur here. So typically for proteins, K is 3 in BLAST. So you can imagine that for each sequence in the database, we store a table of which combinations of 3 amino acid residues appear in that sequence, and we know this for all of the sequences. Now for the query that we present, which could be a new sequence that is not present in the database, BLAST will start by creating the list of these three consecutive residues, of all the combinations of three consecutive residues we have on our query sequence. And this is going to be used to find the best candidates on the database. So those that have similar sequences to the ones that we have in our query. And now BLAST is going to expand and join the segment pair. So if there is, for example, one combination of residues here, one of these words that is similar to one here, it's going to do this match and then try to expand this region by looking at similar residues in the neighborhood here, and the same thing here. And then if these regions are close, it's going to merge them together and thus find this maximal segment pair, which is the pair of a segment in one of the database sequences and a segment in the query sequence that maximizes the alignment score. So it's a local alignment, but note that this is not trying to optimize the alignment by dynamic programming, as we saw before. It's just looking at starting from these matches, like little seeds, is going to expand from there and then see if it can, using this approach, if it can find a large pair of segments from the query matching the sequence in the database that would result in a high score. So this is faster than FASTA because there is no sequence alignment step. It just increases the size of these segments until it finds the maximal segment pairs. And all of these words in the database are pre-indexed. So that information is already computed. It just needs to index the query sequence and then match it to what is in the database. There are some differences in different versions of BLAST. For example, BLAST extends these initial segments as long as the score keeps increasing. If the score starts decreasing, it stops. BLAST 2 has more tolerance. It aggregates neighboring words further apart and so on, and it can deal even with gapped alignment, so it can include gaps in those sequences. And then you can also use MegaBLAST. That's another option that uses larger words. So basically it's much faster if the sequences are similar, but if the sequences are less similar, it probably will miss them, because if you use larger words, larger sequences of residues here in this first step, then the matches are more significant when you find the same word in your query sequence and in other sequences in the database, but then differences between the sequences may prevent those from being matched. So MegaBLAST is better if you want to find very similar sequences in the database, but it doesn't work as well if there is more divergence between the sequences. Okay, one important thing here when we are computing these alignments, these are not optimal alignments, but these quick alignments when searching sequences, is the statistical significance of the alignment. Note that if you have two sequences that you assume are homologous and you find the best alignment by the algorithms we saw previously, there is no reason to concern ourselves with the statistical significance of the alignment, because we are assuming the sequences to be homologous and we just want the alignment to see where they match the best one to the other. But if we are searching in a database, we need to know if the results we got are statistically significant, or if that kind of match can happen simply by chance, because we are looking at a database with millions of sequences and some happen to have that particular score when compared to our sequence. So one important thing in in sequence search is to have this idea of the statistical significance of the results we have. So for this maximal pairs, so the maximal sequences we find that match between the two sequences. If they do not have gaps, we can we can calculate what would be the probability of finding a match with a score greater than X. So this is the probability that by chance, if the sequences are completely random, we are going to find a match to our query sequence with a score greater than X. And the probability will be 1 minus this expression. This K is a scaling factor, M and N are the sizes of the sequences, and there is also this lambda, which is a function of the values in the substitution matrix, because the substitution matrix will determine the score. But in any case, I'm not going to show you how to derive this expression or look at this into much detail, but the idea is that as X increases, the probability that we find by chance a score greater than X will decrease, and we have this tail. It becomes lower and lower as we move on. But we can estimate it, and so we can see if finding a match of score S is significant when we are looking at the database. We can also see how many sequences we would expect to find by chance in this case, in this database, and we can calibrate this using the size of the database. So in the case of FASTA, this expectation was multiplied by the number of sequences. So assuming that we have an equal likelihood a priori of matching our sequence to any of the sequences in the database, the larger the database, the larger the number of matches we expect to find by chance with sequences that are not really related to ours, but just happen to have this score. So in FASTA, we are multiplying this value by the number of sequences by assuming that the probability is the same for matching all sequences. In BLAST, this expected number of sequences of results that we get by chance is obtained by multiplying by the number of residues in the database and divided by the number of residues in the query. That is, if we have a smaller sequence in the query, it assumes that we have more chances of matching at random to something in the database, because a large sequence in the database can match ours by chance in different positions. But in any case, this is a small difference between the two. The idea here is that we can use the probability of matching a sequence by chance with the score greater than X to obtain an estimate of, on average, how many matches we are going to find with a score greater than X, and calibrate this with the number of sequences we have in the database. So if you have, if you search a database and you have an expected number of matches by chance of, say, 10 with the same score as a particular match you found, you cannot have much confidence on that match, because that can be one of the 10 that just are expected to happen by chance. On the other hand, if you find a score that is so high that the probability becomes so low that you would expect only that to happen by chance once in a million or something like that, then you can suspect that this was not by chance, it's really a homologous sequence. Now, these probabilities depend on the substitution matrix, the sizes of the sequences, and so forth. And so these are not very good as a general measure. So this score will not be very good as a general measure of how good the match is, because if you want to convert this into a probability, you need to take into account all of these factors. So a better measure of how good a match is when searching a sequence database is this normalized or bit score that we can compute, which normalizes the score of the match, those segment pairs that are matching, normalizes it by the constant, and so gives us something that we can compare between different matrices and different databases and so forth. So this way we have by looking at the score, we can already have an idea of how significant the match is. So often you're going to see in the results of sequence searches this bit score that gives you an idea of how unlikely it would have been to have this kind of match by chance. So these are problems. So these aspects are more useful if you're actually using sequence search programs. Here in this course we are mostly focused on the ideas on the algorithms. So the most important part here is to understand that, unlike in sequence alignment, when we are doing sequence search, we also need a way of estimating if we are not getting that match by chance and if it is a meaningful match. So for this, it also for finding matches in the database, it's also important to choose the correct substitution matrix. So just to recall from last week, we have these shallower matrices which are based on more highly related sequences, and so they penalize substitutions more and give a greater bonus to conserved regions. These are good for finding matches that are very homologous, or regions that are very homologous, but then will not give you results when you go for more divergent regions. If you use deeper matrices, those that allow for a larger number of substitutions and don't have as much difference between conserved and non-conserved regions, so these are good when you're trying to find matches with a smaller identity, but then it's a problem with the significance of the results and how you can find matches between regions that are not really homologous. So this will depend, the substitution matrix you use also for search will depend on your goals for finding the matches. Okay, one additional problem that we have in sequence search, this may also occur in alignment, but it's especially serious in sequence search, is the presence of low complexity regions. These are regions where we can have repetitions of the same symbol. For example, these Q linkers in proteins, they are stretches of around 20 residues, and then they tend to be very rich in some particular amino acid types. So if you have proteins with these Q linkers, it becomes much more likely to find matches by chance than it would in general in the other regions. So when we are doing sequence search, it is useful to ignore these low complexity regions. For example, in BLAST, there are these pre-processing programs, SEG and DUST, that examine the regions of the sequences, SEG for proteins and DUST for DNA, and mark out or erase, usually by replacing with X or in protein or N in DNA, which means that this can be anything, and so it's not counted for the substitutions. And it does this for low complexity regions. For example, this is for this protein, high complexity regions are these where we have a large diversity of amino acid residues. Low complexity regions have fewer types of residues there and lots of repetitions of the same residue. So these programs, basically the way they work is to consider windows, a sliding window along the sequence, measure the occurrences of different residues, and if this is not close to the general distribution of residues, so if it's too biased towards a small number of residue types, this is considered low complexity, and so it will be ignored when looking at the sequences for sequence search. this is useful because if we were to use these regions with lots of repetitions, we would greatly increase the number of false positives when matching to sequences in the database. There are other options like scaling the substitution matrix depending on the profiles of the query and the other sequences and so on. So you can either hide parts of the sequences or adapt the substitution matrix to account for those repetitions, for that lower complexity of your query sequence. Okay, another way of doing sequence search is this position-specific iterative BLAST, which repeats the BLAST search. You start with an initial BLAST search and you obtain from your initial sequence a set of candidate sequences, so sequences that are apparently homologous to yours. These are local alignments, so they are homologous to parts of the query sequence, and then they will be aligned using a multiple sequence alignment. We're going to look into more detail on this starting from next week, but the multiple sequence alignment is basically an alignment extended to more than 2 sequences. You can have 20 sequences and you align them all in a single alignment. And what this is used for is to compute the substitution matrix differently for each position. So in this case, this would be the positions in the position-specific scoring matrix. For example, position 1 corresponds to residue 7 on our query sequence. This may be because the first residues were not a good match for the homologs found in the database. And now here we have the frequencies on this multiple sequence alignment of the different residues at this position. So basically, if you start with an initial sequence, do a BLAST search, and obtain 50 sequences that may be homologous, you can align those 50 sequences and now count which residues you have on this matching this position of your query sequence. And by counting these and computing the frequencies and so on, like we saw before when computing those substitution matrices, we get this logarithm of the ratio of those probabilities, and this will give us different scores for the substitution of this residue into these different residues, inferred from what we find in this first search with our query sequence. The same thing for the second position, the third position, and so on. So basically now what we have is a set of substitution matrices, each for each position of this region of our query sequence. So now we're going to use this matrix to search again with BLAST, and now we are giving weight to different weights to different substitutions in different positions of our sequence. We now obtain new results because now we are repeating the BLAST search but with a different substitution matrix. Actually, this one is now dependent on the position. We use these new matches to recompute the multiple sequence alignment and compute the substitution matrix again for each position, and then we can repeat this until convergence, until we find no new sequences. So this is good for finding more distant homologs by focusing on conserved regions and using the pattern of mutations we keep finding as an indication of what kinds of mutations we can accept at each different position. So this is very good for finding more distant homologs, but it's also sensitive to the results we get, especially at the beginning, because note that this first search will give us the sequences for the multiple sequence alignment, which we are going to use to compute those substitution matrices for the different positions, which will then determine what comes next and so on. So there will be a positive, a reinforcement effect here, and so it can be very sensitive to the first the first iterations. But in any case, by changing the scores of the substitution matrix in different regions of our query sequence, and depending on the matches that we find, we can now search the database and focus in a way that finds more distant homologs to that particular region. Okay, just a quick overview of other methods that we have that allow us to compare sequences without alignment. So the reason why we use sequence alignment to compare sequences is because we assume that homologous sequences will also be similar sequences, and so if we align the sequences, we have information about how they evolved. But this means that for a reliable alignment, to have this information, we need the sequences to be homologous. If they are not, then alignment is not reliable anymore. It also assumes, when you do the alignment between two sequences, you assume that you have the elements in the same order, because you cannot go back and switch the order of parts of the sequences. So align first two parts and then align one part that comes next in one sequence with another part that comes before in the other sequence, because you cannot do those kinds of loops on the sequences. But sometimes this does not happen. You can have, for example, two proteins which have very similar domains, but the domains do not occur in the same order along the sequence of the proteins. And so in this case, it would be hard to find that match using the classical sequence alignment methods that we saw. So one very simple and intuitive method of comparing sequences is this dot plot. We already saw examples of this in this lecture, but I'm going to detail this a bit more. You represent the positions of one sequence in one axis and the other in another axis. So in this case, we have human and rabbit cytochromes. And then you represent with a dot in every combination where you have identity. So if one residue on this sequence is identical to this residue on this sequence, then you have a dot there to indicate they are identical. You can see some stretches of dots that appear because you have consecutive residues that are identical in the two sequences. And so if you filter by the number of consecutive residues, for example, here I place a dot at the center of every stretch of 3 consecutive residues. So lots of them disappeared, all of this noise, because they were residues that were similar by chance, and that's very common. But now having a sequence of 3 residues identical, it's far less common, but we can still see it here along the diagonal, which means that there are parts of the sequence of human and rabbit cytochromes that are very similar to one another, or identical. In this case, they are stretches of the sequences that are identical one to the other. So this is one way of quickly seeing the similarities or the identities between sequences, and it's not limited by those limitations of sequence alignment. You could find things that were here out of diagonal, which would mean that you have one stretch. For example, if you had one stretch here at the beginning of this sequence matching some part at the end of the other sequence, you could see a line here. Or if you have something at the beginning of this sequence matching the end there, you could have a line here. So it would not be, you could still see that even if the ordering was different. Other approaches can be counting the profiles of the frequency of particular words. So for example, in DNA, you can have the number of combinations will be 4 raised to the size of the word. In proteins, it's 20 raised to the size of the word. So these are the different possible combinations for K symbols in DNA and proteins. And now you can compute for each sequence how many times each particular combination occurs. You can get these frequency profiles for the different sequences, and then you can do some statistical comparisons between these profiles. So I'm not going to go into detail here, but these kinds of computations, Kullback-Leibler divergence, or the Jensen-Shannon divergences, these are all methods to estimate the differences between the different distributions. So we are going to quantify the difference between two distributions, and we take these distributions of combinations of sequences in each of subsequences of these words in each of our sequences, and we use that to quantify how similar or how dissimilar the sequences are. Note that this does not rely on any alignment. It doesn't matter the order in which these words appear in our sequences. It just we are just looking at the differences in the frequency profiles between the sequences. Other methods include using measuring distances between words that occur in both sequences, or how many common substrings we have, matching substrings, and so on. So the basic idea here is that we are looking at statistics of the sequences in a way that allows us to quantify how alike they are without having to actually match everything in order, or match the sequences, or align the sequences. Because if we try to do that, if we try to match everything in order or align it, this only works if the sequences are similar. If they are too dissimilar, then we cannot use those methods. But these other methods that do not depend on alignment can still find common or statistically significant similarities between the sequences, even when we cannot align them because they are too different. So to sum up, this lecture was a bit different because I was not focusing so much on the details of these algorithms, but just to give you a more general idea of these different algorithms for sequence search and for comparing sequences in general. Now, one thing that we have here is that all of these have a different approach to what we saw before, because remember, when we looked at the dynamic programming and the alignment problem, we're looking at ways of finding the optimal solution to that alignment problem. So we have the problem that is very well defined. We need to search all of those combinations, and we have algorithms that search through all the relevant combinations in an efficient way in order to find the best solution. Now, for searching sequence databases, for example, we need a lot more speed, and we cannot really wait for the optimal solution. So all of these algorithms are based on heuristics, on general rules and approaches that tend to work and give us a good enough result for what we want in an acceptable amount of time. So these are the examples we looked at were FASTA and BLAST for doing these searches in sequence databases. And in the case of PSI-BLAST, this is an iterative BLAST where we keep changing the substitution matrix for different positions of our sequence and depending on the matches that we start to find. So this can allow us, this is another approach that does not guarantee any optimal result, but it's a good heuristic to find more distant cousins, more distant relatives of the sequence that we are using for query. We also need to have some estimate of the statistical significance of the results we obtain when searching sequence databases, and we also saw here some other approaches to comparing sequences without having to align them. I recommend that you read at least these sections of the Introduction to Computational Biology textbook. If you want, you can read the complete chapter 4. There are some things there we do not cover in fast alignment for large sequences and so on, but you can read the chapter. You have also, there are in the notes references for these papers, for FASTA, BLAST, and PSI-BLAST, also for the derivation of those formula for the probabilities, if you want to look at this in more detail. And if you want to use search engines for these sequences. In practice, you can look at the NCBI help and documentation pages, but note that this is outside the scope of this course, because here we are not focusing on the practical application of these algorithms, just on the concepts and the notion of the algorithms. So if this is just if you want to use them and to experiment with them.